Hydrogen is the lightest element in the periodic table. When hydrogen atoms are positioned between two electronegative atoms in a very specific way, they form hydrogen bonds. The hydrogen bond is an attractive interaction between a hydrogen atom that is covalently linked to a more electronegative atom, shown here as XH, and another electronegative atom, shown here as Y. The hydrogen has a partial positive charge and acts as the hydrogen bond donor, while the Y forms the acceptor. In biological macromolecules such as proteins and nucleic acids, hydrogen bonds play key roles in stabilizing these molecules and also in their interactions and functions. Let's look at the hydrogen bonds in one of these helices. In this part of the helix, if we hide all the side chains and focus only on the backbone atoms, we see a repeated pattern of N-C-alpha C forming this helical structure stabilized by hydrogen bonds. The carbonyl oxygen acts as the acceptor, while an amino group 4 amino acids away acts as the donor in this hydrogen bond. In the same structure, we can focus on the interaction between the nitrogenous bases of DNA. Each of these GC base pairs form 3 hydrogen bonds between them. In this representation, although the hydrogen atoms are not explicitly shown, their positions are implied. So in each of these hydrogen bonds, the NH forms the hydrogen bond donor, while the O or N forms the acceptors. The distance between the two electronegative atoms in the hydrogen bond is approximately 3.5 angstroms, while the angle, including the hydrogen atom, is approximately 180 degrees. Note that the polarizability, hence the strength of the hydrogen bond, may change with the pH of the environment. Besides the peptide backbone atoms and the nucleic acid bases, the side chains of polar amino acids such as serine, threonine, asparagine, and glutamine, and charged amino acids commonly form hydrogen bonds with each other or with water molecules. Let us examine the interactions of two specific asparagines. The electronegative atoms in these amino acid side chains form hydrogen bonds with the DNA phosphodiester backbone as well as with water molecules.